Welcome back to the deep dive. Our mission today, well, it's not just learning. We're aiming for true mastery, really getting that expert-level grip on the body's most vital logistical system.
Yeah, we're diving deep.
We've got a stack of really specialized sources here, all focused on the nitty-gritty, the microscopic structure, the precise function of blood composition.
And when we say mastery, we really mean it. We're going way past that, high school idea of blood just being red liquid.
Right.
Today, we're looking at it accurately. It's a specialized connective tissue. Yes, it's fluid, but it's packed with these sophisticated cells, the formed elements, all floating in this dynamic protein-heavy fluid matrix. Plasma.
Think of it like the body's ultimate superhighway, maybe, constantly flowing.
Exactly. And in the average adult, you're looking at about, 5 liters?
5 liters, yeah. Constantly moving just one direction, under pressure, doing thousands of things every single second. It's delivering nutrients, hormones, it's the military defense, it's the waste disposal, all of it.
Structurally, though, it boils down to those two main things. The plasma, that fluid part, and the formed elements. The red blood cells, white blood cells, platelets.
Erythrocytes, leukocytes, platelets, got it.
And to really get a feel for how much volume is dedicated to each job, you kind of have to see how we separate them first.
Okay, let's unpack this then. Starting with that visual separation, that heterogeneity, the centrifugation process.
Right. So to do this, it's actually pretty straightforward, but super revealing. You draw the blood, but crucially, you add an anticoagulant first.
Like heparin.
Or citrate.
Yeah, exactly. Something like that. The whole point is to stop the plasma proteins from, immediately starting the clotting process.
Okay, stops the cascade.
Then you take that sample, pop it in a centrifuge, and spin it down really fast. And what happens is everything separates out based purely on density.
And you get these 3 distinct layers, right? A visual breakdown of where the volume is. Let's start at the bottom. The heaviest stuff. Okay, so that dense material packed at the bottom, that's overwhelmingly the erythrocytes, the red blood cells.
Makes sense. They're carrying all that heavy iron in the hemoglobin.
Precisely. That protein cargo makes them sink. And the really critical measurement here is the volume these cells take up. We call that the hematocrit.
And the hematocrit volume is, well, it's huge, isn't it? Like almost half the blood?
It is. We're talking roughly 44% of the total blood volume in a healthy adult is just this one cell type.
Wow.
That tells you straight away, doesn't it? The body's main circulatory effort, its biggest volume investment, is all about oxygen transport, no question.
Okay, so above that big red layer, there's that tiny little band.
Yeah, just a sliver, really. Thin, grayish-white layer. That's called the Buffy coat.
And it's minuscule, like 1% of the volume.
About that, yeah. Maybe 1%.
But small doesn't mean unimportant here.
Not at all. Tiny layer, huge function, because that Buffy coat, that's where you find the other 2 formed elements. The leukocytes, white blood cells, our immune defense, and the platelets, the bits involved in clotting.
Ah. So they're less dense than the red cells.
Much less dense. They don't have all that hemoglobin weighing them down, so they float right on top of the erythrocyte layer.
Gotcha. And then finally the top layer makes up the rest of the volume, about 55%.
Yep, the remaining 55%. It's that clear, straw-colored liquid, maybe slightly viscous. That's the plasma.
Okay, plasma. But wait, you mentioned clotting earlier. We need to clarify plasma versus serum. Right. They're not the same thing.
Absolutely crucial distinction. We got plasma in our centrifuge tube because we deliberately added that anticoagulant before spinning.
So all the clotting proteins, like fibrinogen, are still floating around in there.
Exactly. Fibrinogen's still soluble, ready to go, but inactive. Now, if you don't add the anticoagulant, if you just let the blood sit and clot naturally...
The whole game changes.
Completely. The clotting cascade happens. Those coagulation proteins, especially fibrinogen, get used up. They polymerize, form the physical clot structure.
Okay.
So what's left after the clot forms, that remaining clear, pale, yellow liquid, that's serum.
So serum is plasma minus the main clotting factors.
Mostly, yes. But it's also biologically different in another way. During clotting, the activated platelets release a bunch of important things, including growth factors, right into the serum.
Ah, so serum has extra goodies that plasma doesn't.
Yeah, it gives serum different biological properties, different uses sometimes in labs or therapies. Important difference.
The clarity really helps. Okay, before we dive into the actual molecules in the plasma, let's just quickly recap those main jobs. What are those 5 liters doing constantly?
Well, it all boils down to maintaining balance, right? Homeostasis, the first big one is transport. Obvious, maybe, but huge.
Oxygen, primarily.
Oxygen, yes. Mostly latched onto hemoglobin in the red cells. arterial blood carrying way more than venous, obviously, but also carbon dioxide.
How does CO2 travel? I remember it's a bit more complex.
It is. Some is just dissolved in the plasma. Some binds to hemoglobin, actually different site than oxygen. But the majority, the biggest chunk, gets converted inside the red blood cell into bicarbonate ions, text HCO3 $3, and then shipped out into the plasma. That's the main transport form.
Okay, bicarbonate.
Yeah.
And beyond gases.
Oh, everything else. Metabolites from cells, nutrients absorbed from the gut, hormones traveling from glands to target organs. It's the delivery truck for all of it.
And the flip side of delivery is pickup, right? Collection and removal.
Exactly. Blood is constantly sweeping through tissues, picking up the metabolic leftovers. Things like urea from protein breakdown, bilirubin from old red cells, creatinine from muscle activity.
Waste products.
Yeah, waste products.
Yeah.
And it makes sure they get delivered efficiently to the right places for disposal, mostly the liver for processing and the kidneys for excretion.
Okay, transport, removal.
Yeah.
And the third big one, regulation, keeping the internal environment stable.
This is fundamental. It's about managing the conditions, like distributing heat around the body, helping maintain a stable core temperature.
And chemical balance.
Crucial. Maintaining the very precise acid-base balance, that pH of around 7.4. And also the osmotic balance, making sure the right amount of water stays inside the blood vessels versus leaking out into the tissues. It regulates fluid distribution. #tag2. Deep dive into plasma composition.
All right, let's zoom in on that 55% liquid fraction then the plasma. It's mostly water, right?
Yeah.
But it's this complex solution doing all that work, designed to keep that stable pH around 7.4. What makes it such a perfect medium?
Well, about 92% is water, yes. But what's really fascinating is the constant exchange. See, the smaller molecules in the plasma salts, glucose, urea, things like that, they're basically in continuous equilibrium with the interstitial fluid that bathes all the cells outside the capillaries.
So they're freely moving back and forth.
Pretty much. Which means the chemical makeup of your plasma is the single best real-time snapshot of your entire body's extracellular fluid environment.
So you get a blood test.
You're essentially reading the chemical state of all your tissues indirectly. It's a global report card.
Okay, so 92% water. The other 8% is where the action is. About 7% of that is the plasma proteins. Big category. Let's start with the absolute main player.
No contest there. That's albumin. Makes up a whopping 58% of all the protein in your plasma. It's huge.
And it's made where?
Exclusively in the liver. And you just cannot overstate how important it is. Its number one job, absolutely critical, is generating and maintaining the osmotic pressure of the blood.
Osmotic pressure. Pulling water in.
Exactly. Think of it as a massive concentration of dissolved protein acting like a sponge. It constantly pulls water from the surrounding tissues into the capillaries and keeps it there.
Right.
Prevents fluid just leaking out everywhere.
So if your albumin levels drop significantly, say if the liver isn't working properly or severe malnutrition,
Then that osmotic pull fails. Water isn't held in the vessels effectively, and it starts to leak out into the tissues.
and that causes.
Widespread tissue swelling, that's edema, direct consequence of low albumin.
Wow. Okay, so osmotic pressure is job one, but it does other things too.
Oh yeah, it's a workhorse. It acts as a general, non-specific transport protein. It binds to and carries stuff that doesn't dissolve well in water on its own.
Like what?
Things like free fatty acids, quite a few steroid hormones, essential electrolytes like calcium, and really importantly, many therapeutic drugs. Albumin helps shuttle them around.
Versatile. Okay, moving on. The next big group, the globulins. About 37% of plasma protein, and you said they're functionally diverse.
Very diverse. We usually split them into alpha and beta globulins, which are mostly made by the liver, like albumin. These guys are more specialized transporters.
Okay, examples.
Think of transferrin. Its specific job is binding and transporting iron, which is crucial but toxic if free. Or the various lipoproteins, LDL, HDL, their job is packaging and transporting fats and cholesterol around the body.
So specialized delivery trucks.
Pretty much. And this alpha-beta group also includes many of the essential coagulation factors like prothrombin, which links back to that clotting cascade we talked about.
Right. But then there's another type of globulin. The gamma globulins. Different origin, different job.
Completely different. The gamma globulins are the immunoglobulins. Antibodies.
The immune system.
Exactly. And crucially, these are not made in the liver. They're secreted by specialized immune cells called plasma cells, which you find mostly in lymph nodes and other lymphoid tissues. They're the molecules of adaptive immunity, recognizing and neutralizing specific threats like bacteria and viruses.
Okay, albumin globulins. What's next? About 4% left.
That 4% belongs to the biggest protein molecule in the plasma, fibrinogen. It's enormous, like 340 kilodaltons.
You called it a molecular I-beam earlier, ready to be assembled.
It's a great analogy. Fibrinogen, also made in the liver, is the soluble precursor protein. It just floats around waiting for the signal.
The signal to clot.
Right. When the coagulation cascade gets triggered, fibrinogen gets cleaved, and those resulting fragments instantly self-assemble. They polymerize into these long, tough, insoluble strands of fibrin.
And that forms the mesh.
That forms the structural meshwork of the blood clot. It's the physical barrier that traps cells and stops the bleeding.
Okay. And rounding out the protein picture. The complement proteins, you mentioned them as a defense system.
Yeah, the complement system. It's a whole cascade of proteins, maybe 20 or more. Think of it like dominoes. Activating one triggers the next, which triggers the next.
A rapid response chain reaction.
Exactly. It gets activated by infection or inflammation, and its job is to help destroy microorganisms, sometimes by directly punching holes in their membranes, and also to amplify the overall inflammatory response. It flags things for destruction.
Okay. That covers the 7% protein chunk, but there's still that last 1% of plasma volume. The other solutes, what's in that final percent?
That 1% might sound small, but it's where all the fundamental chemical stability comes from. It includes all the essential electrolytes,
Sodium, potassium, chloride.
calcium, bicarbonate, all those ions. They're not just floating randomly. They are absolutely vital for maintaining the electrical charges across cell membranes. Nerve impulses, muscle contractions depend on this. They regulate that osmotic balance we talked about. And critically, the bicarbonate ions are the key players in the blood's buffering system, maintaining that precise pH of 7.4.
So electrolytes are key for stability. What else is in that 1%?
You've got nutrients being transported, glucose for energy, amino acids for building proteins, vitamins, fatty acids, all the fuel and building blocks being delivered to cells.
Makes sense.
Then respiratory gases. We said most O2 is on hemoglobin, but a tiny bit is dissolved directly in the plasma. And for CO2, while most is bicarbonate, some is also just dissolved gas.
And finally, the waste products again.
Yep, the wastes are in that 1% too. Urea, bilirubin, creatinine, they're just passengers in the plasma at this point, on their way to the liver or kidneys to be removed. So yeah, that plasma is the universal solvent and delivery system for absolutely everything.
Okay, let's pivot now from the fluid matrix, the plasma, to the cells floating within it, the formed elements. And we have to start with the most numerous by far, the erythrocytes, the red blood cells. And here's where it gets, well, really interesting, like you said earlier. Because these cells are so hyper-specialized for one job.
They've basically thrown out everything else.
Exactly.
They're the absolute ultimate example of form following function. We define erythrocytes as terminally differentiated structures. What does that mean?
No nucleus.
No nucleus, no mitochondria, no ribosomes, no endoplasmic reticulum. They've ejected almost all their internal organelles during maturation.
Why? To make more room.
Precisely. To maximize the internal volume for packing in as much of the oxygen-carrying protein hemoglobin as physically possible, they're essentially just flexible bags of hemoglobin.
But wait, no mitochondria. How do they even survive? How do they get the tiny bit of energy they need to, maintain their shape and pumps?
Ah, good question. They rely entirely on anaerobic glycolysis, taking glucose from the plasma and breaking it down without oxygen.
Which is really inefficient, energetically speaking.
Very inefficient compared to aerobic respiration. But it generates just enough ATP to power their essential ion pumps and maintain their structural integrity. The massive downside, though?
No nucleus, no ribosomes means no repairs.
Exactly. They can't synthesize new proteins, can't fix any damage. That limitation is what dictates their relatively short, finite lifespan.
Let's talk about that famous shape. The flexible biconcave disc looks like a little flattened donut without the hole going all the way through. About 7.5 micrometers across.
Yep, around 7.5 mem. And that biconcave shape is just brilliant biological design. It achieves the maximum possible surface area to volume ratio.
Why is that so important?
Efficiency. It means gases, oxygen, and CO2 can diffuse in and out extremely rapidly across the membrane. Plus, it means no internal hemoglobin molecule is ever very far from the cell surface. Faster loading, faster unloading.
Maximum efficiency for gas exchange.
Yep.
And that 7.5 micrometer size is so consistent.
That histologists use it all the time. If you're looking at a tissue section under the microscope and see an RBC nearby, you use its 7.5 min diameter as an internal standard, a built-in ruler, to estimate the size of other cells or structures around it.
The body's own micrometer scale bar.
Exactly. But the shape isn't just about surface area. It's also key to their other essential property, flexibility.
They had to squeeze through tiny capillaries, right? Some are narrower than the RBC itself.
Absolutely. They constantly have to bend, deform, fold almost in half sometimes to navigate the twists and turns and narrow passages of the microcirculation. If they lose that flexibility, disaster. They get stuck, block flow.
Like in sickle cell, we'll get to that. I've also seen pictures where they stack up in larger vessels. Looks like a roll of coins.
Yeah, that's called rouleau formation. They just loosely adhere face to face in slower moving blood. It's normal.
So how do they maintain that incredible flexibility and resilience for months on end? It must be the cell membrane, the plasmalemma.
It is. The erythrocyte membrane is probably the best studied cell membrane there is. Structurally, it's about 50% protein, 40% lipid, 10% carbohydrate by weight.
And some key proteins in there.
Oh, yeah. Two major integral membrane proteins are Ban3 protein, which is an important anion transporter, exchanges bicarbonate for chloride, and glycophorin A.
And those ABO blood type markers we talked about, they're attached to these proteins.
Yes. The complex sugar chains, the glycosylations on the external parts of these proteins, and some lipids, those are the antigenic sites that determine your ABO blood type. They're literally the flags on the cell surface saying, I belong, or I'm foreign.
Okay, so that's the surface. But the real key to their toughness, their ability to bend and snap back, that's underneath the membrane, isn't it? The cytoskeleton.
That's the secret. It's the cytoskeletal meshwork just beneath the lipid bilayer. It's not like the 3D cytoskeleton in other cells. It's more like a 2D lattice or net.
Made of what primarily?
The main component is spectrin. These long, flexible, rod-like proteins link up end to end and side to side. This spectrin network is then anchored to the overlying cell membrane at multiple points through linking proteins, notably one called anchorin, which connects to Ban3.
So it's like a flexible reinforcing cage just under the surface.
Exactly. This submembranous meshwork is what gives the membrane its strength, maintains the biconcave shape, and crucially provides the elasticity needed to withstand the constant squeezing and shear forces in the circulation for, well, about 120 days. It's built for punishment.
And inside that bag, it's just packed wall to wall with hemoglobin. That tetrameric protein gives it the characteristic red color, or acidophilia in stains. And its job is simple: reversibly bind oxygen,
Forming oxyhemoglobin. And it can also bind some CO2, forming carbaminohemoglobin. That reversibility is key. Binds strongly in the lungs, releases easily in the tissues.
But even the best-built structure wears out eventually, especially with no repair crews. So that 120-day lifespan is pretty firm.
It is. Around 120 days, things start to fail.
How does the body know? Is there like a timer on each cell? How does the spleen pick out the old ones?
It's not really a timer, more like accumulated damage. Over those 120 days, the spectrin meshwork starts to degrade slightly, the ion pumps become less efficient, maybe some proteins get oxidized, the surface carbohydrates might change subtly. The cell just becomes less flexible, less efficient.
So it starts to fail its squeeze test in the spleen.
Basically, yeah. These subtle changes tag the cell as senescent or defective. And specialized macrophages, phagocytic cells, primarily located in the cords of the spleen, but also in the liver, called Kupffer cells there, and in the bone marrow, recognize these old or damaged RBCs and gobble them up, recycling the parts.
A tightly regulated system, but when it goes wrong, when that concentration isn't right, we get disease. Too few RBCs.
That's anemia. Below normal concentration means not enough oxygen carrying capacity. Leads to symptoms like lethargy, shortness of breath, fatigue, looking pale, pallor. Very common.
And then there are problems with the structure itself, the classic example being sickle cell anemia.
Right. That's caused by a single gene mutation homozygous recessive affecting one amino acid in the hemoglobin protein. Under low oxygen conditions, this mutated hemoglobin clumps together. It polymerizes inside the cell.
And that distorts the shape.
Dramatically. It forces the flexible biconcave disc into a rigid, deformed crescent or sickle shape.
And those rigid cells cause huge problems.
Huge. They can't squeeze through capillaries easily. They increase the blood's viscosity, making it thicker. And they block small vessels, that's called vasoocclusion, which is incredibly painful, and damages tissues downstream due to lack of oxygen.
Nasty. And the opposite problem: too many red blood cells.
That's erythrocytosis, sometimes called polycythemia. It can be a normal adaptation, like if you move to high altitude, your body makes more RBCs to compensate for center air.
Or it can be a disease state, right? A type of blood cancer.
Yes, polycythemia vera is a myeloproliferative disorder, but either way, adaptive or pathological, Having too many RBCs makes the blood significantly more viscous. Thicker blood puts a huge strain on the heart trying to pump it, and paradoxically can actually impair flow in the smallest capillaries. All right, let's shift gears from oxygen delivery and waste removal to, well, defense, the armed forces. The leukocytes, or white blood cells.
The immune cells.
Exactly. Now, these are way more complex and varied than RBCs. But here's the single most important concept to grasp right at the start. While they're floating around in the bloodstream, leukocytes are generally spherical, mostly non-motile, and basically inactive.
So they're just patrolling, waiting
Like-
for a signal, hitching a ride in the circulation.
Precisely. Their real job, their entire function, fighting infections, cleaning up debris, initiating tissue repair, all of that happens only after they get the signal to leave the bloodstream and migrate out into the surrounding connective tissues.
That's where the action is.
That's where the battle takes place. Once they get the exit signal, then they become actively modal, kind of amoeboid, and head towards the problem.
Okay. And we classify these defenders based on their appearance, right?
Yeah.
Especially what's inside them, granules.
Yep. The main classification is based on whether they have prominent, specific cytoplasmic granules and also the shape of their nucleus.
So group 1.
Group 1 are the granulocytes. Think of these as the rapid response chemical warfare specialists. This group includes neutrophils, eosinophils, and basophils.
What defines them?
They all possess distinct specific granules in their cytoplasm containing different functional molecules. And characteristically, their nuclei are weirdly shaped polymorphic, often segmented into multiple lobes.
Then they don't live long.
No, they're terminally differentiated, meaning they won't divide again. They have very short lifespans, usually measured in days. And they primarily rely on glycolysis for energy, which is handy in damaged low oxygen tissues.
Okay, granulocytes, group 2.
Group 2 are the agranulocytes. These guys lack those obvious specific granules. Their cytoplasm looks more uniform, maybe slightly grainy, but not packed with distinct blobs. This group includes lymphocytes and monocytes,
And their nuclei?
usually simpler shapes, spherical or maybe kidney bean shaped or indented, but generally not lobulated like the granulocytes.
No, you said specific granules. Does that mean they both have some other type?
Ah, good catch, yes. Both granulocytes and agranulocytes contain some azurophilic granules. These are essentially the cell's standard issue lysosomes containing digestive enzymes. They just don't define the cell type like the specific granules do in granulocytes.
Got it. So the key thing is that they have to get out of the blood vessel to do their job. This exit process, diapetesis, or extravasation.
Both terms are used, diapetesis. It's this incredibly elegant, multi-step process. It doesn't happen just anywhere. It specifically occurs in the walls of small veins called postcapillary venules, usually right near the site of injury or infection.
And it's triggered by local signals.
Absolutely. It's a precisely coordinated 5-step cascade. You really need to understand these steps to see how the defense is mobilized.
Okay, lay it out for us. Step 1.
Step 1, cytokine release. It starts locally in the tissue. Maybe a resident macrophage bumps into some bacteria or tissue cells get damaged. These activated local cells immediately release chemical alarm signals, pro-inflammatory cytokines. Key ones are interleukin-1, IL-1, and tumor necrosis factor alpha, TNF, TEL-alpha.
So the tissue screams for help chemically. What happens next?
Step 2, select an insertion. Those cytokines drift the short distance to the nearby postcapillary venule. The endothelial cells lining the inside of that vessel detect the cytokines and react instantly. They rapidly shuttle adhesion molecules called p-selectin from inside the cell, where they're stored in little packages called Weibel-Palade bodies, onto their luminal surface, the side facing the blood flow.
So the inside wall of the blood vessel suddenly gets sticky, specifically for leukocytes.
Exactly. Which leads to step 3, rolling. Leukocytes, especially neutrophils, which are usually the first responders, have specific carbohydrate molecules on their surface that act as weak ligands for p-selectin.
Ligands, like a key fitting a lock.
Sort of, but a very weak temporary lock in this case. Think of it like Velcro. The leukocyte briefly sticks to the p-selectin, gets tugged by the blood flow, unsticks, tumbles a bit, sticks again.
So it slows them down dramatically. Instead of whizzing by, they start rolling along the vessel wall.
Precisely. This rolling adhesion is crucial because it gives the leukocyte time to sense other signals and prepare for the next step.
Which is?
Step 4, firm adhesion. While the leukocyte is rolling, those same cytokines, IL-1, TNF-alpha, stimulate it to activate different, stronger adhesion molecules on its surface called integrins. At the same time, the cytokines tell the endothelial cell to express the ligand for these integrins, a molecule called ICM1.
So stronger glue molecules appear on both surfaces.
Right. When the activated integrin on the leukocyte grabs onto the ICM1 on the endothelium, that's it. It's a firm, stable bond. The rolling stops. The leukocyte is arrested, stuck tight to the vessel wall. And importantly, the cytokines also signal the endothelial cells to temporarily loosen the tight junctions between themselves.
Creating a little gap for the exit.
Exactly. Which brings us to the final step, step 5, diapedesis and chemotaxis. The arrested leukocyte, now activated, becomes amoeboid. It sends out little projections, probes the junctions between the endothelial cells, finds that loosened spot,
And squeezes through.
squeezes, or sometimes it's described as leaping right through the gap, out of the blood vessel and into the underlying connective tissue. Diapedesis is the physical act of crossing the vessel wall.
Okay, it's out.
Okay.
Now what? Does it just wander around?
No, it's guided. Once in the tissue, it follows a chemical trail. The site of injury or infection is releasing other signaling molecules called chemokines. The leukocyte follows this chemokine gradient, moving towards the highest concentration. That directed movement in response to a chemical signal is chemotaxis. It leads the cell right to the target.
Incredible sequence. Okay, let's meet the granulocytes properly now, starting with the absolute workhorse, the most common one. The neutrophils. They make up, what, 50 to 70% of all circulating leukocytes?
Yeah. The vast majority, sometimes called PMNs, right? Polymorphonuclear leukocytes.
Why polymorphonuclear?
Because of their nucleus. It's highly variable or polymorphic, typically segmented into anywhere from 2 to 5 distinct lobes, all connected by thin strands of chromatin. Looks quite distinctive.
And size-wise.
A bit bigger than red blood cells, usually around 12 to 15 micrometers in diameter.
And their main job, first responders.
Absolutely. They are the first wave of defense to arrive at a site of acute bacterial infection. Their primary role is phagocytosis, engulfing and destroying bacteria and cleaning up debris. They follow those chemotactic signals right to the bacteria.
Okay, and their killing power comes from those granules we mentioned. You said granulocytes have specific granules plus the standard azurophilic ones. Let's break down the neutrophils' arsenal.
Right. They have two main types of cytoplasmic granules packed with different weapons. First, the azurophilic primary granules.
These are the lysosomes, basically.
Essentially, yes. They are larger, denser, stain dark purple-blue, azurophilic. They contain a potent mix for killing and digesting. Key contents include myeloperoxidase, MPO, a really critical enzyme we'll come back to, lysozyme, which breaks down bacterial cell walls, and defensins, small proteins that poke holes in bacterial membranes. Think of these as the heavy-duty demolition tools.
Okay, primary granules. What about the specific ones?
The specific secondary granules are generally smaller, less dense, and stain faintly pink or lilac. These contain a more specialized toolkit.
Like what?
Things like enzymes that degrade the extracellular matrix, such as collagenases, which helps the neutrophil move through tissue. They also contain various antimicrobial peptides and proteins that get delivered into the phagosome. And importantly, they contain components that get inserted into the cell membrane during activation, like receptors.
So a two-stage weapon system. How do they actually use this to kill bacteria? You mentioned MPO and something called the oxidative burst.
Ah, yes, the oxidative burst or respiratory burst. This is the neutrophil's nuclear option for killing. It's incredibly powerful.
Walk us through it.
Okay, so the neutrophil engulfs a bacterium into a vesicle called a phagosome. Then both the primary azurophilic and specific secondary granules fuse with this phagosome, dumping their contents inside. Proton pumps also acidify the phagosome interior.
Okay, acid bath plus enzymes.
Right, but simultaneously, the neutrophil undergoes a dramatic metabolic shift. It suddenly starts consuming oxygen at a huge rate. Enzymes in the phagosome membrane, particularly one called NADPH oxidase, use this oxygen to generate highly reactive oxygen species, ROS, like superoxide.
First, superoxide anions, then another enzyme converts superoxide into hydrogen peroxide.
Hydrogen peroxide, like in the brown bottle.
Sort of, but generated right there in the phagosome at high concentration. Now, here's where myeloperoxidase, MPO from those primary granules, comes in. MPO takes the hydrogen peroxide and, using chloride ions that are also pumped in, converts it into hypochlorite.
Hypochlorite. Isn't that basically bleach?
It is the active ingredient in household bleach. So the neutrophil creates this highly toxic bleach solution right inside the phagosome, along with other ROS. It's an incredibly effective killing mechanism. Plus, another protein called lactoferrin is released, which binds up iron, essentially starving any bacteria that might survive the chemical onslaught.
Wow. Acid, enzymes, bleach, starvation. Brutal.
Extremely effective against most bacteria.
But all that chemical warfare must take a toll on the neutrophil itself. You said they rely on glycolysis. Can they survive this? They can survive in anaerobic conditions, which is useful in damaged tissues with poor oxygen supply. But the whole process is energetically demanding, and they are designed for a short, intense life.
How short?
Very short. Their half-life, just circulating in the blood, is only about 6 to 8 hours. Once they enter the tissues and get activated, they typically only last for 1 to 4 days before they undergo programmed cell death apoptosis.
Such a high turnover rate. What happens when they die?
Well, at the site of a significant infection, you get a massive accumulation of these dead and dying neutrophils, along with the bacteria they've killed, liquefied tissue debris, and extracellular fluid, that semi-liquid yellowish material. That's pus. Mostly dead neutrophils.
A battlefield graveyard. And if this powerful killing system, like the oxidative burst, fails due to a genetic defect,
that causes serious problems. For example, there's a condition called chronic granulomatous disease, CGD, where there's a defect in the NADPH oxidase enzyme complex.
so they can't make superoxide or hydrogen peroxide properly.
Exactly. The oxidative burst is severely impaired. They can still phagocytose bacteria, but they can't kill them efficiently, especially certain types. This leads to recurrent severe bacterial and fungal infections, often forming granulomas, clusters of immune cells trying to wall off infection they can't clear. Shows you how critical that burst is.
Okay, neutrophils are the bacterial specialists. Let's move to the next granulocyte. Eosinophils, much less common, only 1 to 4%, but they look quite striking, don't they, with those bright pink granules?
They really stand out on a blood smear. Bright pink or reddish granules, intensely acidophilic or eosinophilic, meaning eosin-loving, eosin being the pink acidic dye. They typically have a characteristic bilobed nucleus, looks like 2 lobes connected by a thin strand.
And their specialty isn't bacteria, it's parasites.
Especially multicellular helminthic parasites, parasitic worms. That's their main claim to fame.
How do the granules help with that? They're too big to phagocytose, a whole worm.
Right, so the eosinophil releases the contents of its specific granules onto the surface of the parasite. Those large oval granules have a unique internal structure, a flattened crystalline core.
A crystal inside the granule, what's it made of?
That core is packed with major basic proteins, MBP. These are highly cationic, arginine-rich proteins, and they are what cause the intense pink staining, acidophilia. MBP, along with other granule contents like eosinophil peroxidase and various toxins, are directly damaging to the outer layers of parasitic worms. They basically try to dissolve the worm from the outside.
Wow. So targeted chemical attack on large parasites. Do they do anything else? You mentioned allergies earlier.
Yes, their numbers, a condition called eosinophilia, often increase significantly not just during parasitic infections, but also during allergic reactions. They seem to play a role in modulating inflammatory responses. They can release various cytokines and chemokines, and they also phagocytose antigen-antibody complexes, perhaps helping to clean up after an immune reaction.
So anti-parasite and inflammation modulation, where do you typically find them?
They are abundant in the connective tissue underlying epithelia, especially in the respiratory and digestive tracts, places where parasite entry or allergens are common. Also found at sites of chronic inflammation, like in the airways of people with asthma.
And things like steroids affect them.
Dramatically. Corticosteroids, which are powerful anti-inflammatory drugs, cause a rapid decrease in the number of circulating eosinophils, partly by blocking their release from the bone marrow.
Okay. Neutrophils, eosinophils. That leaves the rarest granulocyte, the basophil. Less than 1%. Almost never see them on a smear.
They are indeed the rarest, about the same size as neutrophils, 12-15 Vim. Their defining feature is the presence of very large, coarse granules that stain intensely dark purple or blue basophilic.
And these granules often hide the nucleus.
They often completely obscure it. The nucleus itself is irregular or bilobed, but you can rarely see it clearly because of these huge, dark granules packed into the cytoplasm.
What's in those granules that makes them stain so darkly, and what do they do?
The intense basophilia comes from highly sulfated glycosaminoglycans, GAGs, within the granules, particularly heparin, which is a potent anticoagulant. But they also contain massive amounts of histamine.
Histamine, the allergy molecule.
The very same. Histamine causes vasodilation, widening of blood vessels, and increased vascular permeability, leakiness. The granules also contain other mediators of inflammation, like platelet activating factor, eosinophil chemotactic factor, which attracts eosinophils, and phospholipase A, which helps generate leukotrienes, another class of inflammatory molecule.
It sounds incredibly similar to mast cells, those cells found in connective tissue that are famous for releasing histamine and allergies. Are basophils related?
Functionally, they are very closely related. They really supplement the function of mast cells. Both basophils and mast cells have these characteristic metachromatic granules, meaning they can stain a different color than the dye used. And critically, both cell types have high-affinity receptors on their surface for a class of antibody called immunoglobulin E, Ig.
IgE, the antibody associated with allergies.
Exactly. And this is where basophils and mast cells play a central role in immediate or type 1 and hypersensitivity reactions, like hay fever or food allergies or anaphylaxis.
Anaphylactic shock, how does that work?
Okay, first exposure to an allergen, like pollen or peanut protein, causes your B cells to make IgE antibodies against it. These IgE antibodies then circulate and bind tightly to those IgE receptors on the surface of basophils and mast cells, essentially arming them.
So they sit there, coated in IgE, waiting.
Then, upon a second exposure to the same allergen, the allergen molecules bind to and cross-link these IgE antibodies already sitting on the cell surface.
What does cross-linking trigger?
It triggers immediate massive degranulation. The cell basically dumps its entire load of histamine and other mediators instantly into the surrounding tissue and bloodstream.
And if this happens systemically, all over the body at once?
The sudden, widespread release of histamine causes massive vasodilation everywhere. Blood pressure plummets catastrophically. Airways can constrict. This systemic reaction is anaphylactic shock, and it can be rapidly fatal if not treated immediately, for example with epinephrine. Basophils are key players in initiating this dangerous cascade.
All right, we've met the granulocytes, the neutrophils, eosinophils, basophils with their specialized granules and often short dramatic lives. Now let's turn to the other main group, the agranulocytes. No obvious specific granules, starting with the cells that orchestrate the really sophisticated immune responses. Lymphocytes. They're pretty common, right? 20–40% of leukocytes.
Yes, they are the most numerous agranulocyte and second most numerous leukocyte overall after neutrophils. Morphologically, especially the small ones just released from the bone marrow or thymus, they look quite simple. They're often about the same size as a red blood cell, maybe 7–8 m. Their defining feature is a large, typically spherical nucleus that's packed with very densely clumped chromatin stains. Very dark. And there's only a very thin, scant rim of pale blue cytoplasm visible around this dominant nucleus. Minimalist design.
But they can get bigger.
Oh, yes. When lymphocytes get activated, say by encountering an antigen they recognize, they can enlarge significantly, maybe up to 18 m. The nucleus becomes less condensed, and the cytoplasm increases as they prepare to divide or produce effector molecules, so size can vary.
Functionally, though, they're incredibly diverse. We can't tell the different types just by looking at them under a standard microscope, can we?
Not reliably. Morphologically, they look very similar. We have to use specialized techniques, usually involving antibodies that recognize specific protein markers on their cell surface, the CD markers, cluster of differentiation. And these markers define the major functional subtypes. Exactly. We classify them into B lymphocytes, B cells, which mature in the bone marrow and are responsible for producing antibodies, T lymphocytes, T cells, which mature in the thymus, and natural killer NK cells.
And the T cells have subtypes.
Yes, the main ones being helper T cells, which usually express the CD4 marker plus DD and help coordinate immune responses, and cytotoxic T cells, which usually express CD8, CD plus D, and are responsible for killing infected or cancerous cells directly.
You mentioned T cells maturing in the thymus. That's unusual, isn't it? Most blood cells mature in the bone marrow.
It's unique among circulating leukocytes. B cells, granulocytes, monocytes, RBC precursors, platelet precursors, they all complete their maturation within the bone marrow. But T cell precursors leave the bone marrow and travel to the thymus gland, located in the chest, for their crucial maturation and selection process. This thymic education is essential for ensuring they can recognize foreign threats but don't attack the body's own tissues.
Fascinating. And their lifespans, you said granulocytes live for days, lymphocytes.
It varies enormously. Some newly formed lymphocytes might circulate for only a few hours or days before migrating into tissues or undergoing apoptosis if they don't find their specific antigen. But others, especially memory lymphocytes, both T and B cells that are formed after an infection or vaccination, can survive for many years, potentially even a lifetime, providing long-term immunological memory.
That's the basis of immunity, long-lived memory cells.
Precisely. However, because lymphocytes can divide and circulate widely, cancers involving them, lymphomas, usually in lymphoid tissues, or leukemias, often involving circulating cells, can be particularly dangerous and prone to spreading throughout the body.
Okay. Lymphocytes handle the adaptive immunity. What's the other main type of a granulocyte? The monocytes.
Monocytes. These are the largest type of leukocyte you'll normally see in peripheral blood, typically 12 to 15 micrometers, sometimes even larger.
And they have a distinctive nucleus, too.
Very distinctive. It's large, but usually not spherical. It's typically indented, often deeply looking C-shaped or kidney bean-shaped. Their chromatin is also less densely clumped than a lymphocyte's, appearing more lacy or folded.
And the cytoplasm?
It's more abundant than in a small lymphocyte, and it's typically grayish-blue, sometimes described as having a ground glass appearance. It does contain some fine eosinophilic granules, lysosomes, which contribute to that slightly granular bluish-gray look, but again, no large specific granules like a neutrophil.
So what's the monocyte's job? Is it active in the blood?
Like other leukocytes, mostly no. Monocytes are essentially circulating precursor cells. They spend a relatively short time in the blood, maybe a few days,
Before doing what?
before they follow signals to leave the bloodstream using that same diapetesis process and migrate into various tissues throughout the body. Once they enter a tissue, they differentiate
Differentiate into what?
into the various long-lived resident phagocytic and antigen-presenting cells of the mononuclear phagocyte system.
Ah, so they become other cells, like macrophages.
Exactly. They become the tissue macrophages found in connective tissue, Kupffer cells in the liver sinusoids, osteoclasts that remodel bone, microglia, which are the resident immune cells of the brain, and specialized antigen-presenting cells like dendritic cells in the skin and lymph nodes. Almost every organ has its population of these monocyte-derived cells.
So monocytes are the precursors, the reinforcements, for this whole network of tissue watchmen and garbage collectors.
That's a great way to put it. They're constantly replenishing these tissue populations.
And their presence or accumulation signals certain types of inflammation.
Yes. While neutrophils dominate the very early stages of acute inflammation, like the first 24 hours of a bacterial infection, the sustained recruitment and accumulation of monocytes migrating out of the blood and differentiating into macrophages is characteristic of chronic inflammation.
Why is that significant?
Because if the inflammatory stimulus persists, like in chronic infections, autoimmune diseases, or reactions to foreign bodies, you get this continued influx of monocytes. The resulting large population of activated macrophages, while trying to help, can also cause significant ongoing tissue damage over time through the release of enzymes and cytokines. Chronic inflammation is often driven by this mononuclear cell infiltrate.
Okay, we've covered red cells, granulocytes, agranulocytes.
Yeah.
That leaves one last formed element.
Yep.
And it's not even really a full cell, is it? The platelets or thrombocytes.
That's right. They are unique. They're very small, only about 2 to 4 micrometers in diameter, much smaller than RBCs. And critically, they are non-nucleated. They are essentially membrane-bound fragments of cytoplasm.
Fragments of what? Where do they come from?
They originate from enormous cells found in the bone marrow called megakaryocytes. These giant cells extend long, branching cytoplasmic processes right into the bloodstream within the bone marrow sinusoids.
And bits just break off.
Exactly. The tips of these processes pinch off or fragment, releasing thousands of these small, membrane-enclosed packets of cytoplasm of platelets directly into the circulation.
Incredible production line. And they don't live long either.
No, their lifespan is quite short, only about 10 days on average. So there's constant production needed to maintain the normal count, which is quite high, typically between 150,000 and 400,000 platelets per microliter of blood.
And their job is all about damage control, right? Stopping
Yeah.
bleeding, hemostasis.
Precisely. They are the first responders to vascular injury, plugging leaks, and initiating the blood clotting cascade. To understand how they do this, we need to look at their internal structure, even though they're just fragments.
Yeah, break it down. What's inside a platelet?
Structurally, we often describe 2 zones. There's a lighter staining outer or peripheral zone called the hyalomere, and a darker staining central zone packed with granules called the granulomere.
What's in the hyalomere, the outer part?
The hyalomere contains a crucial cytoskeletal element, the marginal bundle. This is a ring of microtubules and actin filaments running just under the cell membrane around the platelet's circumference.
What does that do?
It acts like a scaffold, maintaining the platelet's characteristic discoid, lens-like shape when it's inactive and circulating. The hyaline also contains two important membrane systems.
Which are?
First, the open canalicular system, OCS. This is a network of deep invaginations or channels of the surface membrane that extend throughout the platelet interior. Think of it like interconnected tunnels open to the outside.
What's the point of that? Increased surface area.
Massively increased surface area, yes. It allows for rapid uptake of factors from the plasma, but more importantly, it provides a route for the extremely rapid release degranulation of the platelet's internal contents when it gets activated. Stuff stored deep inside can get out quickly through the OCS.
Okay, OCS for release. What's the second membrane system?
The dense tubular system, DTS. This is derived from the megakaryocyte's smooth endoplasmic reticulum. It's a separate, internal network of tubules, and its main job is to store high concentrations of calcium ions. Calcium. That's often a trigger for cellular processes, isn't it? Absolutely critical in platelet activation and the coagulation cascade itself. The DTS is the internal calcium reservoir.
Okay, that's the hyaline. Now the granulocyte, the central part, that's where the actual payload is.
Exactly. The granulocyte is darker because it's densely packed with various types of storage granules, plus mitochondria for energy, and glycogen particles, fuel storage. There are two main types of granules we need to know.
Go on.
First, the delta granules, also called dense bodies. These are small and appear very electron-dense in electron micrographs. They contain small molecules crucial for activating other platelets and causing vasoconstriction. Key contents are ADP, ATP, and serotonin.
ADP is a big platelet activator, right?
Huge activator. Release of ADP from the first few platelets sticking to a wound site recruits many more platelets to the plug.
Okay, delta granules, the other type.
The alpha granules. Alpha dollars. These are larger, more numerous, and less electron-dense. They contain a wide variety of proteins involved in adhesion, aggregation, coagulation, and vessel repair.
Examples.
Important ones include platelet-derived growth factor, PDGF, which stimulates smooth muscle and fibroblast proliferation to help heal the vessel wall, and various proteins that promote clotting, like platelet factor 4, fibrinogen, yes, platelets store some too, and von Willebrand factor.
So delta granules for initial activation, alpha granules for reinforcement and repair, all packed into this tiny fragment.
Ready to deploy instantly upon activation.
Which brings us to the process itself, hemostasis. Stopping blood loss. You said it's a sequence of events.
Yes, typically described in 4 overlapping steps. It starts immediately when a blood vessel is damaged.
Step 1.
Step 1. Primary aggregation. The damage exposes collagen fibers in the vessel wall that are normally hidden from the blood. Platelets have receptors that allow their outer coat, the glycocalyx, to adhere directly to this exposed collagen. This initial adhesion causes the first few platelets to stick to the site of injury, forming a loose initial platelet plug.
Just a temporary patch at first.
Right. But this adhesion triggers the platelets, leading to step 2, secondary aggregation. The platelets that just stuck undergo activation, they change shape, become spiky, and crucially, they degranulate, releasing the contents of their delta and alpha granules, especially ADP, and another activator called thromboxane A2, which they synthesize.
And this released ADP acts as a chemical alarm.
Exactly. ADP and thromboxane A2 bind to receptors on other nearby circulating platelets, activating them and causing them to stick to the platelets already in the plug. This creates a positive feedback loop, rapidly recruiting more and more platelets, enlarging and consolidating the platelet plug. This is secondary aggregation building a bigger plug.
Okay, the plug stops the immediate outflow. But it still just clumps platelets, right? Not very strong.
Correct. It's fragile. It needs reinforcement. That's step 3, blood coagulation. This is where the plasma proteins get involved, the cascade we talked about earlier. Triggered by factors released from the damaged vessel wall, tissue factor, and also by components exposed or released by the aggregated platelets themselves, like platelet factor 3 on their surface and platelet factor 4 from alpha granules. These initiate the complex sequence of enzymatic reactions involving the coagulation factors, like prothrombin converting to thrombin.
And the end result of that cascade?
The end result is the enzyme thrombin converting soluble fibrinogen from the plasma and some from platelet alpha granules into insoluble fibrin polymers.
The mesh.
The fibrin mesh. These fibrin strands polymerize all around and through the aggregated platelets, essentially weaving a strong, stable net that traps red blood cells and more platelets, physically reinforcing the plug. This stable structure of fibrin, trapped cells, and platelets is the definitive thrombus, or blood clot.
Okay, bleeding stopped, clot formed. What's the final step?
Step 4. clot retraction and removal. Once the clot is formed, the platelets within it actually help to compact and stabilize it further. They contain actin and myosin filaments, like muscle cells but less organized, which contract, pulling the fibrin mesh tighter and squeezing out residual serum. This is clot retraction.
Makes it smaller and denser?
Yes. Then, over the next few days, as the underlying vessel wall is repaired, stimulated partly by factors like PDGF released from platelets, the clot needs to be removed so normal blood flow can be restored.
How does it get dissolved?
That's mainly the job of another plasma protein system involving plasmin. An inactive precursor called plasminogen gets incorporated into the clot as it forms. Later, activators, like tissue plasminogen activator, TPA, released slowly by endothelial cells, convert plasminogen into the active enzyme plasmin. Plasmin is a protease that specifically digests fibrin, breaking down the clot meshwork, a process called fibrinolysis.
So, a beautifully orchestrated process. Plug formation, reinforcement with fibrin, then controlled removal once healing occurs. But it's a delicate balance, isn't it? Clotting only where needed, not elsewhere.
Extremely delicate, and we exploit that balance medically. Think about common anticoagulant or antiplatelet drugs.
Like aspirin. How does that work in this context?
Aspirin is a classic antiplatelet drug. It works by irreversibly inhibiting an enzyme called cyclooxygenase, COX, inside platelets. This enzyme is needed to synthesize thromboxane A2.
And thromboxane A2 is important for
Secondary aggregation and vasoconstriction. So by blocking thromboxane A2 production, aspirin reduces the ability of platelets to aggregate and activate each other, thus inhibiting clot formation. Other agents might target ADP receptors or interfere with the coagulation factor cascade itself. It's all about carefully tipping that balance. #JohnAlaid, conclusion and final takeaways.
Wow. Okay, we started this by defining blood not just as red liquid, but as this specialized fluid connective tissue. And we've gone through everything. The complex soup of plasma proteins, the specialized structures within every type of leukocyte, right down to these tiny enucleated platelet fragments that orchestrate clotting. It feels like we've really achieved that deeper level of understanding, that mastery.
I think so. And if you really connect all those dots, the bigger picture that emerges is just stunning, the sheer integration. Think about it. The precise engineering of the RBC's spectrin skeleton, allowing it to bend, specific osmotic power of albumin holding fluid in, that intricate five-step dance of diapedesis allowing WBCs to exit on cue, the multi-layered cascade of coagulation.
So it all works together.
It's this unified, incredibly complex, yet somehow seamlessly functioning system, operating constantly, second by second, purely to maintain that stable internal environment homeostasis. Every tiny structural detail we discuss serves that ultimate macro function.
Right. Let's try and crystallize the absolute key takeaways for someone aiming for that expert-level grasp. What are the non-negotiables?
Okay, for plasma, remember, it's the transport matrix, and albumin is king for maintaining osmotic pressure, preventing edema. Fibrinogen is the soluble precursor for the clot's fibrin mesh.
For RBCs.
Erythrocytes. Think terminally differentiated enucleated biconcave discs, optimized for gas transport, packed with hemoglobin, flexible due to that spectrin cytoskeleton, and a strict 120-day lifespan before removal by macrophages.
Leukocytes.
White blood cells. Remember, they're functionally effective only after leaving the circulation via that crucial five-step diapedesis process.
And the granulocytes specifically.
Neutrophils are the most abundant, the primary bacterial phagocytes using granule enzymes and that powerful oxidative burst. Eosinophils target large parasites, helminths, and modulate allergic responses. And the rare basophils are loaded with histamine and heparin, closely linked to mast cells and central to immediate hypersensitivity and anaphylaxis.
And the granulocytes.
Lymphocytes, B, T, NK cells, orchestrate adaptive immunity, with T cells uniquely maturing in the thymus and memory cells providing long-term protection. Monocytes are the large precursors that circulate briefly before migrating into tissues to become the diverse cells of the mononuclear phagocyte system, macrophages, etc., key players in chronic inflammation.
And finally, platelets.
Platelets, tiny enucleated fragments derived from megakaryocytes, essential for hemostasis, forming the initial plug via aggregation, primary, secondary, and then initiating the coagulation cascade leading to fibrin formation and a stable thrombus. Remember their OCS and DTS systems and delta alpha granules?
It really is a system of constant turnover, constant defense, constant logistics, which I think leads perfectly into our final provocative thought for you, the listener, to ponder.
Yeah, think about this. We talked about the incredibly short lifespan of neutrophils. They're the frontline infantry, but they only last 1 to 4 days out in the tissues before they have to undergo apoptosis, given how numerous they are and how vital they are as first responders.
Imagine the scale of production required.
Exactly. Consider the sheer, mind-boggling continuous volume of new neutrophil production happening deep inside the bone marrow, and the equally massive programmed removal of the old ones every single hour of every day, just to maintain that defensive readiness. What are the hidden, ongoing, energetic, and metabolic costs to the body of managing this immense daily turnover, billions upon billions of these highly specialized, short-lived cells? Reflect on that silent, relentless factory operation the next time you just think of blood as blood.